Click for IB Biology, the mechanism of DNA replication. There's a blank PDF of this presentation down in the description, along with a quick quiz with answers. Some channel links will appear at the end to other videos in this sequence. There's actually an AI prompt as well that you can use to further your own studies. This model is appropriate to standard level, although higher level students should be completely familiar with this before proceeding to the next level. So let's take a look. Here we have the DNA double helix. This could either be an event which in eukaryotic cells, this would take place inside the nucleus, but of course in prokaryotic cells that would be the nucleoid, and there being no nucleus, this would take place in the cytoplasm. So the first major item to consider is that the DNA molecule has to be unwound. And this is the action of DNA helicase. And what DNA helicase will do will be to separate the two polynucleotide strands by breaking the hydrogen bonds between their complementary bases in the center. You can see in the image that we have the 5' to 3' chain on this side, whereas the 3' to 5' antiparallel strand is on this side. So the helicase will progressively work towards up through the DNA molecule in this direction. If we take a look to the right hand side, we can see that the helicase enzyme has progressed up to the CG complementary base pairing. But behind this, what has happened is that nucleotides which are in the nucleus, which were generally, these are referred to as free nucleotides of the form A, T, G, and C, adenine, thymine, guanine, and cytosine. These are available. The free part refers to them not yet having become complementary base paired. So as you can see, on this side we had adenine, and a free nucleotide of the type thymine will come in here and hydrogen bond, complementary base pairing A to T. At the next nucleotide, which is G on this side, that will attract a cytosine nucleotide, and they also will hydrogen bond. Then the next one is thymine, which will attract in an adenine base with the nucleotide, and they will complementary base pair as well. This is also happening on this side of the molecule, but for space-saving reasons I didn't show that. And so the helicase continues to unzip the DNA molecule, breaking the hydrogen bonds, and behind that the bases come into position, complementary base pairing, not only on this side, but on this side also. To complete the process, it's necessary for a second enzyme, which is known as DNA polymerase, to complete the sugar phosphate backbone along the new nucleotide strands. So on this side here, the bonds are being made where I'm pointing the arrows to at these positions here, joining the 5' carbon to the 3' carbon, and this repeats along this backbone. Similarly, the same event occurs on this side with the DNA polymerase joining up the backbone on this side, forming the covalent phosphodiester bonds between the sugars and the phosphates. At the end of this process, notice that the base sequences or the base pairs in both of the chromosomes is the same, both of the DNA molecules, AT here, AT here, and so forth, all the way down the bases. The difference being is that each of the new molecules, and these molecules are identical, of course, in their base sequence, contains half of the original and then a new polynucleotide chain. So this side and this side have acted as templates for the construction of the new polynucleotide chains. And the chains are accurately built through the complementary base pairing. Since they contain half of the original and half of a new strand, this is called semiconservative DNA replication. And so the DNA molecule has copied, and these are shown in the space-filling models shown below. In cell division, what actually happens is that these two identical copies will be held together by a small piece of protein called a centromere, and they will form a kind of an X shape, which we can see in the image here. This classic image of a replicated DNA molecule is shown here, and this is actually one DNA molecule on this side and the identical copy on this side, held together at this point here by a centromere. These are actually referred to as a pair of chromatids, and we will return to this image when we look at the process or the events of cell division. DNA replication mechanism involves unzipping with the helicase, complementary base pairing, and then sealing of the sugar phosphate backbone by the polymerase enzyme. Higher level students can now follow the link to the higher level mechanism, and then there are other links in the playlist to other parts of this topic.